which is a very important part of our research as ecologists and plant ecologists. So, for instance, she collaborates with farmers to integrate the ecological principles into future suitable farming systems and leads research programs, other research programs related to biodiversity and food system, to nature, inclusive society, and she's guiding policymakers and politicians with her research. So I'm sure that we are all looking forward to her talk, and we are delighted to have her sharing her knowledge with us today. So let's give a very warm welcome to Professor Liesje Mommer. Enjoy the talk, and please, Liesje, the floor is yours.
Thank you so much. Good morning, everyone. It's a pleasure to be here. It's a great honor to be here. The last time I visited IBC, that was in Vienna, and then I was still a PhD student. So it's really an honor to be here and have this stage today. I will talk to you about exploring the underground, and I was willing to talk about only about the treasures we are trying to dig up in the soil. And I was about to talk about you to my journey in the underground. However, after hearing Sandy's excellent talk of the opening, on Sunday, I felt I also need to speak up more about my other job in Wageningen University, about biodiversity, because I think it's really critical that we are aware about the importance of biodiversity laws for the functioning of our ecosystems, but also for the importance of biodiversity for society, basically for us. So, as already introduced, I have initiated and I also lead the Wageningen Biodiversity Initiative that aims to create a network of scientists, students and stakeholders to work on bending the curve of biodiversity loss. So what was it that made me so inspired by the talk of Sandy last Sunday? It was specifically this graph that was published in New Phytologist last year. And I'm really happy to see that climate change is so prominent in this figure. And so yesterday and the day before were the two hottest days on Earth in human history. The hottest days on Earth. Climate change is with us. So it's really important that we are aware, that as botanists, that we are aware of that, because it's a really important topic for us to study. It's a really important for society that we work on it. So I was happy to see that climate change is featured so prominently in this outlook for the future for botanists. Actually, I was also a little disappointed. It was good to see that there is diversity mentioned in these figures, so diversity and biodiversity even. But I was shocked to not see that biodiversity loss is in there. So we as botanists, knowing so much about the diversity of plants and how evolution develops, we seem to have a blind spot, at least in this picture, for the importance of biodiversity loss. So maybe we are building trees of life where we are already also losing so much unnoticed. So my point is, and this is a very important cartoon from the times of COVID, we should not forget about biodiversity loss. So in this cartoon, it's even shown that biodiversity collapse, may even be worse than climate change. Actually, I think it does not matter to talk about what is worse. For me, climate change and biodiversity loss are two sides of the same coin, and we really need to work all together to really stop this. So this is my really important point for today, for my lecture. I want to really convey that bending the curve of biodiversity loss is also a responsibility of us botanists. And this really should be our daily task. So I have 2 teenagers at home, and every night during dinner time, I think, did I do enough? Did I do my ultimate best to work on this? Because if we don't do anything, you know, we'll be on this gray line. You know, if I feel the heat, if I don't see plants around in this area, I think we are just not on a pathway towards a prosperous future. I see a lot of conservation talks here in this wonderful congress, but will not be enough. We will have to work on more sustainable production and more sustainable consumption. And this is, you know, we botanists, we know how important that is. And also Sandy already said 80% of our diet consists of only 4 species, you know. We, you know, this community, knowing so well what a wonderful diversity of plants we have, we can help. We can help diversify our cropping systems. So I really hope that in the next version of this paper, biodiversity loss is central. There are also 2 other topics that I missed in this graph, and that is planetary health and plant health. So health is here in the corner. But I thought planetary health and plant health are also critically important, and I will talk about that for a large part of the talk today. Planetary health. This picture I always find really shocking because it really shows how much we are crossing planetary boundaries. And if you would like to have some numbers for it, you know, so the way we produce our food really is responsible for 80% of global deforestation. It contributes to greenhouse gas emissions, and also it really degrades our soil. And if the soils are degraded, you know, we will, you know, we are tempted at least to clear more land for agriculture. And that again drives to diversity loss in terrestrial, but also in freshwater systems. So planetary health is at risk, and for me this picture really shows that very clearly. We have to ensure that we will come and live more within the planetary boundaries, if it's about emissions of greenhouse gases, but also if it's about protecting biodiversity. So both species and also genetically. That is really critical. That will really depend, you know, our future of our children will depend on that. Planetary health is also visible for me in this picture. Again, about how we grow our food, and especially in the Netherlands and in Europe, you know, the way, you know, the amount of pesticides that we use is becoming a topic of planetary health. Also about human health. So as evidence is accumulating that the massive amount and also the diversity of pesticides are used is affecting our health. It's also related to plant health, because even with these intense spraying, you know, we are not able to keep our plants healthy. So maybe this system, the way, you know, these monoculture systems are intrinsically vulnerable to pathogens. And actually that is not a new idea, but that was already being said by Sir Elton in the mid 1900s. He already said, or he already wrote in his book, that it seems that agricultural monosystems are much more sensitive to pathogens than diverse natural systems. And this notion led to a huge debate among ecologists. So the idea is that if you have an increased species richness, your disease risk will be reduced. And as you can see, I wrote some key papers where that has been debated. But the good thing when preparing for this talk today, I came across a very recent paper in Nature. It's a meta-analysis understanding the global drivers for infectious disease. And this meta-analysis is about diseases in plants and also in animals and humans. And it's of course, this is a botany congress. It's really important that it also includes plants. And this is the key result. There's a lot to say about this, but the key point that I want to make is that biodiversity loss is a key driver of disease. So biodiversity loss in general leads to increase of disease. It's the most important driver even if you compare to things as climate change or habitat loss. So I think that is really a critical take-home message. Biodiversity loss is not only important for ecosystem productivity, etc., it's also important for it's also important for disease. But, and this is where my research comes in, it's not that the species richness is a magical trick. It's not just biodiversity, because indeed the general pattern is that biodiversity is decreased, disease is decreased with biodiversity, but not always. Sometimes biodiversity, increasing biodiversity increases even disease. So it's important to just dig a little deeper into this topic. So it's more than just species numbers. And the question that I will be answering also in a huge grant that I received recently is, can we predict based on community composition the risk of disease? And this is critically important if you want to transform agricultural systems from monoculture systems to more diverse systems. Because if we want to increase diversity in agricultural systems, we better do it right. We take the best species combinations. So what can we learn from nature? So what can we learn from ecology to use in agricultural systems? So I take you to biodiversity experiments, and here you see the Jena biodiversity experiment. And in all these blocks, you know, it's a block of 20 by 20 meter, and there are different plant communities grown. So sometimes there is only one species, but also 2, 4, 8, 16, up to 60 species in a randomized block design. And a lot of people are working and taking samples in this experiment. And in these experiments, so if you see an increase of plant species richness, it has been shown that biomass has been increased. And it is not only in the biodiversity experiment in Jena where this is the case, but also in experiments in the US, in Minnesota, and also in my very own Wageningen. Plant species richness goes together with increased biomass production. So this is really a key lesson of 20 years of biodiversity research. And when I came into this field 20 years ago, the main hypothesis was that this plant diversity is enhanced, of that productivity is enhanced by plant diversity because of differences in niche differentiation below ground. So a monoculture, then in a monoculture, like here, all species go for the blue resource. And if you would grow a mixture, then there's a diversity of resources can be used. And if a diversity of resources can be used, then also more biomass can be produced. So if the resources are explored more completely and more efficiently, then that would drive increased productivity. However, my research, but also of others, has shown that niche partitioning alone cannot drive this biodiversity productivity relationship. So this key hypothesis of 20 years ago cannot explain the full story of why we find increased productivity in these systems. So the field was in need of another hypothesis. And there was an alternative hypothesis formulated. That is that pathogens are diluted with diversity. And that is also visualized in this cartoon. You know, in a monoculture species, you only have, you know, the pathogens that are really specialized on this monoculture species, and in a more diverse community, you have a higher diversity of pathogens, but you have less pathogens of that specific red pathogen that is there. So with diversity, you have the idea is that you find a higher diversity of pathogens, but a lower abundance of specific pathogens. And that is also what we showed here. At least we showed how plants respond to this. And now the field has been learned a lot from plant soil feedback experiments. And these experiments consist of two stages. So there's a conditioning stage where you take inocula from the field, from different monocultures, and you then basically train, you sort of activate these communities. So what you do is you grow the plant species that were on your inoculum. You really grow that to create a more bulk soil of that soil. So you really have active and trained plant pathogens, or pathogens, but also other microbes in these pots. And then the real experiment starts. So we call that the test phase. And what we did in this specific experiment is we had these 4 different monocultures, and we also had a mixture soil where we diluted basically. So we mixed it proportionally. And what we then did on all these 5 soil types, we grew also 4 monocultures and the mixture plant species. And this is basically what we find. But I first show you, just some pictures of some of the grasses. So if a plant species is growing on its own soil, it really grows badly. You can really see, after 6 weeks, these plants are still seedlings. They are really suffering. They are not growing well. If they grow in the mixture soil, they grow much better. You don't need statistics to see that plants are more green here and are more lively and look much more better. So what is the final conclusion of this experiment is that, and I talk you to this figure on this is the plant community. So and then the second row here is the soil community. So the first bar is how a monoculture plant species grows on its monoculture soil. So this is the first bar is how a plant grows on its own soil. The second bar is how a monoculture grows on the mixture soil. So where we diluted by 1/4 the own pathogens. And the third bar is how a plant species grows on its soil, on all soils but its own soil. So how, for example, and I can go back. So, for example, how a pink plant species grows on red, green, and blue. And what you very clearly see is that the less of its own soil community is there, the better this plant species grows. So, yes, so this is really consistent with pathogen dilution. However, I was a little disappointed with this. Not with the outcome, but I thought as long as we don't really target the pathogens, we cannot know whether this pathogen dilution hypothesis really works. So what I try to do is to open the black box of soil organisms. And I will show some of the experiments that we are doing in this line. So, we run our own biodiversity experiment. In Wageningen we had 16 plant species, 8 forbs and 8 grass species. And we had them in monocultures, and from the 16 species, we also created 90 different 4-species mixtures. So we decided to focus on having different communities rather than having a whole gradient of diversity. So and what we did is we sampled the roots of individual plants in this experiment. We sampled 16 monoculture species and also the mixtures. We also wanted to sample these mixtures, the 4-species mixtures, but we sampled only after 4 years, and some species were really rare, were really not so abundant in these species mixtures. So we had to decided to focus on 8 focal species. So although we have investigated all the monocultures, we had to focus on the most abundant focal plant species in these mixtures. So what we did is we extracted a DNA from the roots. We targeted the fungal communities, and we characterized the pathogen species. So, and in total we had about 900 OTUs for fungi, and of these 900, more than 50 appeared pathogenic, but these 50 pathogenic ASVs really related to 9 pathogenic species. And we thought that is already quite a lot, and we only focused on the most abundant pathogens that were really present in more than 5% of all samples, and then we only had 3 pathogens left. So and these are the results of the monocultures, and the species names in bold are the ones that we will also focus on in the mixtures. But here you see here you see basically the results of our 3 pathogens, and you see that we found 1 generalist pathogen, that is Rhizoctonia solani. We found that's the yellow one. We found that across all plant species. And we also found Paraphoma chrysanthemicula, that is a specialist on forbs, and really I would even say it's a specialist on Asteraceae species, and we also found a specialist for grass species. So we can now focus on these 3 main soil-borne pathogens, and the exciting part is how do they, how is their behavior, or how is their abundance if you compare monocultures versus mixtures. And we expected, of course, this dilution. We expected to find dilution with diversity. Yeah? So the dark bars are the monocultures, and the light bars are the mixtures, and we expected diversity. However, if you just compare the forbs and the grasses, we don't see that. There is no dilution effect, no general dilution effect in forbs for this pathogen. There's even a significant increase in grasses. So we thought maybe we should focus more specifically on the different species, but there we find the same pattern. We do not see a very significant dilution effect. Actually, we find increased abundance of this specific pathogen in poor host species. And we basically find the same if it's about the grass pathogen. We do not see evidence for the dilution effect. So again, there is a there is a significant but increase of these pathogens in the poor host, in the non-host, in the forbs. And also for the generalist pathogen, we do not find a general dilution effect. So also here, we do not see a dilution with diversity. So I think I've made my point that it's not only about diversity. So we thought maybe we should have a closer look into the relationship between whether the forb grass community drives these pathogens. And I can be very short on this. We did not find that the forb grass ratio would drive the patterns for any of these 3 pathogens. And then we thought maybe we should calculate a community weighted mean based on the monoculture averages, and then maybe we can understand the pathogen abundance. Also, that was not such a clear effect. But what gave an effect, what was a significant driver of pathogen abundance, was the presence of specific plant species. So some high quality hosts had a significant effect on the presence of some of the pathogens. And specifically this Achillea mellifolium was very important. If this was in the plot, so if this Achillea mellifolium was present in the plot, then for Centaurea iacea, here the abundance was really significantly increased, and also for Sanguisoba officinalis, for the first 3 species here, we really saw increased pathogens. Interestingly, however, if you took the same analysis, but for the generalist pathogen, you see that if this pathogen, or if this plant species is present in the plot, then we find here the opposite. So Centaurea iacea experiences increased abundance of the forb specialist, but reduced pathogen abundance of the generalist. So apparently there is the abundance of the presence of this plant species also changes some of the interactions between some of the pathogen species. So things are becoming more complicated. So I think that my first step, what we took from this experiment, is that it's indeed more than species number. So sometimes it's biodiversity can also enhance pathogen diversity. So we really thought we really should dig deeper into this community composition. And with the experiment that we did, even though we had 90 mixture plots, we felt we were, reaching the limits of what our experimental design could do. So we did another experiment in the same experiment. But we grew now phytometers, and we added. We have been growing in all plots. We have been growing phytometers of 2 different species, one of an Asteraceae, so Lycanthemum vulgare, and the other a grass species, Arenatrum elaceus. So what we did is, again, we sampled roots to get out the DNA of the fungi. We investigated the resident species, the phytometers, and also the bulk soil of each plot. And we basically wanted to know whether the growth or the pathogen communities in the phytometers would be affected by the resident community, so basically by the surrounding community of these plots. Indeed, we did that, and we also, So this is what we found here. You see the fungal communities of the phytometers, so of the 2 species that we had, and you see the blue dots. These are the Lycanthemum plots of the Lycanthemum phytometer, so the Asteraceae, and the orange ones are the grass phytometer. And it's very clear that these different species harbor different fungal communities. That is not a surprise. That's what we know, that these different phytometers have different species. But what is also interesting, and that is what you see here with this, basically with this wave. Here you also see that there is a significant effect of forbs and grasses over this effect. So this means that the fungal community assembly in these phytometers is driven by the neighbors. And it's both an effect of soil and a direct effect of these phytometers on, a direct effect of the residents on the phytometers. So what I wanted to say is that the forbs and phytometers built up different specialized fungal communities, and that the community where they're in really determines their fungal community composition. And then we thought we have to be more clever than grass forbs or presence absence of some of the species. So we thought we should use plant phylogenetic distance as a proxy for community composition. And then the hypothesis is that if there's a higher phylogenetic distance, the pathogen abundance of these phytometers will be lower. And this is also visualized here. So in a monoculture, the distance is really much smaller. So the red distance is much smaller than it's here. Well, than it's here in this mixture plot. So what did we find? So what we basically wanted to know is whether the phylogenetic community distance, plant species richness, and the biomass of the community, of the original community in this experiment really affects the composition of the fungi in their roots and also their growth. And we did a structural equation modeling approach. And first I showed the data for the Asteraceae seedling for Leucanthemum vulgare. And what I want to highlight, so in this kind of analysis, there's always so many different relationships that you can find. What I think is really important to highlight for the story of today is that there is a direct effect of the phylogenetic community distance on the build-up of the specific pathogens. Oh, wait. So the phylogenetic community distance affects how much pathogens are in the soil, and that affects also the specific pathogens of the phytometer. And if there's more specific pathogens here, then there is more specific pathogens in the phytometer. So there is a transmission pathway from the diversity of the community towards the phytometer. And it's important to show that this is not the case for the grass phytometer. Oh, here it's here. So you see far less correlations, and there is no specific effect to the specific buildup of the pathogens. Then, and that was rather disappointing, and we will have to work more on this. We did not find a significant relationship with the phytometer growth. So the abundance of the specific pathogens, on these phytometers alone did not affect the growth of these phytometers, at least in the 3 months that they were in the field. So what I wanted to say is that there's more than species numbers, and we used phylogenetic distance as a proxy in our study, but we will have to work more on this, and also we have to work more on how this really relates to plant growth in the field. And of course, there will also be competition for light, for example, because if diverse systems also have higher biomass, and the competition for light will be more intense. So entangling these different processes needs some more time, but I also have a grant to do that. Yeah. Okay. I also said already that these phytometers have been in the field for only 3 months, so that is really short. So, and we also specifically knowing that the build-up of diversity productivity relationships needs time, so that increase over time. We know that these, from these different experiments in the US, in Germany, and also in Wageningen, that build-up of productivity relationships need time. So what we did in the Jena experiment, we had the opportunity to really test that, and also to just test how the fungal communities change over time. And what we specifically wanted to know is whether fungal community changes over time are consistent with dilution effect theory. Again, we sampled plots. We had old plots and young plots. We sampled monocultures and the species mixtures. And there we could not, in the Jena experiment, we could not investigate specific species. So we focused on forbs, grasses, and legumes. And again, we were able to investigate, from all the sequences, we investigated the different fungal guilds, including the pathogens. And this is the result that we find. And here you see the young plots, and there you see the old plots, and you already see, the different. So these are the compositions of the fungal communities, and you see that they diverge over time. So older communities are more dispersed over time. So they are more specialized. So the specificity of these communities increase over time, and you both see this in the young and in the old plots, and the gray area is showing, basically the shade of the other. So you can really see that these dots are outside of this gray area, which is the projection of the young plots. So the older plots are more different, and they are more they are more dispersed. And if you then also focus only on the pathogens, you see that with age, you see that with diversity, here's the diversity. You see that at least in the young plots, the diversity increases. So with diversity, you have an increased diversity of pathogens. Yeah. With diversity, you have an increased diversity of pathogens in the young plots. But after 20 years, if there has not been a possibility of pathogen transmission, then you would expect, then you see that the black line is not positive, but is increasing, so that changes over time. You also see that the pathogens are lost from the system. And also, if you investigate the relative abundance, you see that the relative abundance increases slightly in the beginning, but that really the abundance of the pathogens is reducing over time. This is also compatible with pathogen dilution, and I'm really looking forward also to really do in these two type of experiments, to repeat the experiment with the phytometers, to really be able to show how this is growing, evaluating over time. So what I wanted to say is that I hope I've shown you that diversity is also important for how we think about disease risk in plant communities. It's just more than a magical number of species, and that we have to dig deeper into community composition. I also, one step that we already made in my team is that phylogenetic distance can be a useful proxy to work with, and also that the fitness effect of the plants need to be, that they need more time than at least the 4 months that we had in the time with the phytometers. Why is this all important? And I already made a little note about this in the beginning of my talk. our agricultural systems are so monocultural, you know, and we really need to diversify agriculture. And we are currently working on this a lot in Wageningen. And basically we just are growing here. Here you see an example of how we are doing strip cropping agriculture. And we try to have 6 up to 12 different crops on a field rather than having one. But the way we combine species is a little bit trial and error. It's also based on what is agronomically feasible. But we are really working on also on finding how we can use diversity to reduce the pathogen loads and reduce the need for pesticide use. That is what I wanted to tell this morning. I would like to thank you, and I also would like to thank you for your attention. I would like to thank the organizers for giving me the opportunity to speak here. And I also would like to thank my lab members, my current and my former lab members, for all the work they did. Because digging in the dirt is really hard work, and revealing these hidden interactions is tough. So I'm really grateful for their continuous work and effort to reveal these interactions. Thank you so much.
Many thanks, Liesje, for this wonderful lecture. And now it's time to questions, if you have. Yeah, here.
Well, thank you for a wonderful talk. So, first question I've got really is, when these experiments are done in mixtures, the plants are sown so that they're relatively closely intermingled. And just wondering how that relates, first of all, to so a natural system where you've got localized dispersal, etc. And one might argue about, okay, you have to be concerned about the animal side of it with dispersers of seed and pollen, and the agricultural side of it, where we're not accustomed to growing a cabbage next to a sunflower.
So the first question is about the way we do biodiversity experiments, and I fully agree. It's a in that sense it's not a natural system. We really manage the we manage the communities. We also do weeding. That is also that can be quite disturbing, specifically in monocultures. So that is not natural. Still, it showed us very mechanistically how things work, and people are trying to for example, to translate to how that relates to natural systems, and there are large parallels and differences. And I think that is only useful. So I think that is a main critique to diversity experiments. They are not nature. No. They are not nature, I fully agree, but they help us understanding these relationships. For the agricultural side, this is just exciting. Maybe it's a very good idea to grow cabbage next to sunflower, but we don't know, right? And what we are just combining now is at least what I know from my grandfather. And that was more than 50 years ago. So I think it's important to start rethinking if we can do better, because the way we are now is also a dead-end street with the massive amount of pesticides that we are using. So maybe we should start growing a cabbage with sunflower.
There is one there.
Yeah, me. Okay. Hi, I'm here.
Yeah.
So thank you for officially opening the black box of the soil in this conference. So you really focus on pathogen dilution, and that's very cool. But what do you think about the general effect by different plant species that generally improve the presence of beneficial microbes, both fungal and bacterial species? Because it's quite known that more plants recruit more different taxa of beneficial microbes.
This is very interesting. So we know what we have learned from this biodiversity experiment is that so if you have higher species richness, you have a higher microbial biomass. That includes fungi and bacteria, and I just decided to start with the pathogens. So with the pathogens, often it's the nematodes or the fungi. So that's why I decided I first want to go there. But now you ask me, I think it's really interesting to see how pathogens also interact with all the others, because if there are more saprophytes in the soil, maybe there's less space for pathogens. So I think I will be moving also into these directions. It's very interesting, but it's also, it's so diverse in the soil. So I'm always amazed that if I'm working in these biodiversity experiments, there we think working with up to 60 species is already massive, and then below ground, there's 900 or whatever, thousands of species and taxa to work with. So that is really a challenge. But it's very important. I think there is also interactions among these fungal guilds and among other groups. It's really important. Yeah. Thank you so much for the question.
Thank you.
Thank you for your talk. You made an important point that biodiversity is not just counting things, that community interactions include important functional groups. If I'm thinking about the push-pull agricultural system in Kenya, one of the most important components of that agricultural community is desmodium. It's the nitrogen-fixing legumes. They're used as a push crop because they're repellent to root pathogens of corn, the witchweed, right, which is a parasitic plant, but also repellent to herbivores. And I think what's being revealed in those studies now that they're in their 15, 20 years down the road is that it's about soil conditioning as much as a reduction of pathogen load or herbivores. Did your experiments or the Jena experiments include any nitrogen-fixing plants that might have a disproportionate effect on soil conditioning, for example?
Oh, this is an interesting question. So there has been a huge debate also on these biodiversity experiments on the role of legumes. In the beginning, people thought this biodiversity effect was due to the role of legumes. So in Wageningen, we did an experiment without legumes, and we thought, okay, we won't find a biodiversity experiment, but we did. So we had no legumes in the system, and we did find basically similar patterns as systems with legumes, but at a slower pace. So I think the role of legumes is critical. I don't know so much whether it's the legumes and their conditioning, how that is. There is exciting work though that also legumes and legume systems also. And I don't remember the papers by heart, but there is evidence that these legumes also increase free-living bacteria that are fixing legumes. So it's really interesting to look into that. It's a very good question. Thank you.
Hello. I have a question here in the back.
Yeah.
Hello. Thank you for that great talk. It's really exciting. In your first story, you mentioned that the effects seem to be mediated especially by some high-quality hosts. And I was wondering if you could expand on what you meant by high quality, and if that was specifically from the pathogen perspective, or if there were traits like leaf nitrogen or root nitrogen or something like that that made them high quality. And in general, what you think about a trait-based approach. I mean, maybe instead of or in addition to the phylogenetic diversity approach that you looked at. Thank you.
Thank you. That is a great question, and that was also the basis of my thinking of my ERC grant. So you really nailed it down. So that is really what we will be investigating in the next 5 years in this huge grant. I think host quality. So my perspective is that we have been focus too much on this dichotomy between hosts and non-hosts, and I think there's a whole gradient in between. So indeed, some species are really high quality hosts from the perspective of a pathogen, and there are some maybe that can just sort of maintain the pathogen, and that is really critical also in having the system. So I think bringing more perspective from host to non-host, on the contribution of hosts to the pathogen dynamics or to the pathogen fitness, I think that is really one step. And I think we have this, there is this gradient of fast, slow growth, and this is also due to, so you either have fast acquisition or slow acquisition, but the slow acquisition comes with being better defended. So I think there is lots to be discovered. I don't know yet, and I hope to update you in the next conference on what we found there. It's a very good question, very exciting.
Well, thank you for letting me ask a second question. So I'm thinking that the kinds of experiments you've done with the sorts of species you've got, they're really most relevant to what we would call grasslands. And when we're thinking about that, perhaps we should be thinking about the production of meat in pastures that are often been converted, not necessarily to monocultures, but sometimes monocultures. We think about some parts of the world where, for instance, subtropical areas where the African buffel grass has been replacing the native species, or if you look at the Central Valley of California, where nearly all the native grass species have gone and been replaced by introduced annual grasses. We've got a complete changeover in those systems there. They're both non-native species and in some cases much simpler in biodiversity. And then we have the issue of the huge biomass of mammals that are of domestic mammals that are using vast tracts of the earth for meat production. So I'm wondering whether you could comment on the interplay of these different things and how your experiments might relate to that.
Yeah, I know that I work in these grassland systems to really learn how such a system operates in nature, where all these mechanisms are in full swing. So that is why I work in these natural grasslands that are also close to my home. I really want to learn from a system where all these mechanisms are at stake to translate that to arable farming. So what can we learn? And I think it will be, there will be many steps before we get there to have arable systems that have, at least here in Europe, that also include a more perennial crop. So this is one side of my story. I know there is, my work has relevance for management of grasslands, but I want to really go, before I retire, I want to have a new roadmap for how to do arable farming. So this is one side. And my other point, and also from the biodiversity initiative that we did, I think we really have to go towards more plant-based diet. So we will have to drastically reduce our meat production and our meat consumption. And I have to confess, I was not there at the conference dinner yesterday, but I explored the most wonderful vegetarian vegan restaurant in Madrid. I can highly recommend it if you have to go out tonight for dinner.
What's its name?
I'm sorry, but we need to stop here. I'm very sorry. You can continue discussion later. Thank you very much, Liesje, for this talk.